Ladies and gentlemen. I am Marwa Zafarullah, a graduate student in genetics and genomics, and member of Dr. Lucas Kumar Laboratory at UC Davis. I am studying the mechanism behind the rapid evolution of centromere proteins in Arabidopsis thaliana. It would be my pleasure to introduce the first speaker from UC Davis faculty, Dr. Daniel Runcie. He is an assistant professor in the Department of Plant Sciences. Dr. Runcie in his lab focuses to study the evolution of plant life history, evolution of gene regulation, quantitative genetics and statistics, computational model of plant physiology, and gene networks. He completed his PhD at Duke University and was a postdoc scholar in the UC Davis Department of Evolution and Ecology before joining the faculty in 2015. Please welcome Dr. Daniel Runcie.
All right. Well, thank you. Thank you for that introduction, and thanks for the invitation to speak today. So today I'm going to talk about something that's been an interest of mine for several years, and it's a project to try to think about how to do genetics, or quantitative genetics in particular, on maybe the whole suite of traits of an organism, rather than just one trait at a time. So as we've heard lots about today, quantitative traits are really central to our understanding of genetics and to our goal of improving varieties by breeding. As we've heard about, these are things like size or height or yield or root morphology. And when you have a population of individuals like this, sort of central questions that you might ask are, how much of this variation that we see among individuals is genetic? Of that genetic variation, what genes underlie it? And then in an applied sense, can we use that information to select the next individuals to take forward in a breeding program? So all of these questions are really statistical, and by far the most used statistical model in quantitative genetics is this linear mix model, which we've heard about some today. So the linear mix model basically breaks down the variation in a trait, which I'll call Y, into components that can be associated with specific loci or treatments, and then random effects like genetic background or G by E or microenvironment and things like that. So this model is really the foundation of a lot of quantitative genetic methods. QTL mapping, GWAS, genomic prediction, they're really all just the same model. And there's been a lot of development and work in this model recently, especially with the explosion of whole genome genotype data. But I think it still has some really fundamental limitations. And the one that I'm going to talk about today is that, to a large extent, this model is really limited to working on one trait at a time. And I think Chris Talk gave some sort of background on this earlier. I think that's really limited the type of genetics that this field is focused on. So what I'm going to try to do today is try to convince you that, at least in some cases, thinking about the genetics of whole suites of traits together is important, and I'm going to present a method that I'm working on to try to extend this linear model framework to really any number of traits. So there are two main contexts that I think that a joint analysis of a suite of traits together is important. And the first one is when you just have a set of traits that you're interested in, several different traits that you're interested in, say yield and flowering time and drought tolerance altogether, or the performance of a line across multiple environments, say in multiple regions. When you have a set of traits like this, you might be interested in, is there a shared genetic basis to the variation in each of these traits? Are there pleiotropic genes that affect multiple traits? From the breeding side, can we optimize all these traits at once? And the fundamental issue here is that if there are shared genes, and thus the traits are genetically correlated, then if you select on one of them, you'll get changes in the other one due to indirect or correlated selection. And the way around this, or the way to account for this, is to measure or estimate this correlation directly using a multi-trait model. Now that's one case. The other case is where that trait that you're interested in is inherently, or at least can't be characterized by a single number. It's inherently multidimensional. These might be things like shape or color or the whole growth curve of an organism through time. Here, any single measurement is almost meaningless by itself. And if you want to do selection, you really have to think about the whole suite of characteristics at once. Now, we know in principle how to deal with this, and with what's called the multi-trait linear mixed model. However, this model is really not very widely used, and for several reasons. One, that it's really computationally and statistically challenging, and it's also can be hard to interpret the output. And there really aren't very many existing methods that do this well. There are only a few programs out there can even accept 10 or 15 traits together. However, today, there's a lot of interest in high-throughput phenotyping, which are technologies that can measure hundreds or thousands of characteristics of an organism at once. And for example, here with gene expression, with RNA-seq, we can measure the expression of tens of thousands of genes in every sample. Or imaging technologies, here are pictures of Arabidopsis plants that were taken every hour over several months, or like what Chris talked about earlier. We can get data on the growth trajectories or morphology throughout time. And so this is what I mean by big traits, not tall plants, but big data sets. And so I think that in both of these cases, thinking about these traits jointly has a lot of potential. On the imaging side, the size of a plant on any particular day is maybe less interesting than the dynamics of changes in growth curves through time. Or with gene expression, any particular gene might be interesting in itself, but I think there's also the opportunity to learn about the functioning of cellular pathways or developmental processes, which you can look at when you look at the whole suite of traits together. So now briefly, the challenges of doing this. On the statistical side, as I mentioned, when you have multiple traits, the thing that's important is the correlation among them, particularly that genetic correlation. And if we have 2 traits, there's one correlation. Now, if we jump to 10 traits, now there's 45 different correlations we have to estimate among pairs of traits here. And with each correlation you add, you're going to need more data to be able to estimate these effectively. So now, if we go to 10,000 traits, say, which is still smaller than you get in an RNA-seq data set, there would be about 50 million genetic correlations you have to estimate. And so I think you're going to need at least hundreds of thousands, if not millions of samples to be able to effectively estimate all those parameters. And I just don't see that happening. That's bigger than human genetic data sets, really. So that's one sort of fundamental statistical problem. On the interpretation side, though, say we could estimate 50 million associations among traits. What would you do with that? How do you begin to make sense of 50 million numbers? For me, at least, I think thinking about or trying to do that is sort of missing the bigger picture. In these big data sets or big data, the goal really shouldn't necessarily be to find that needle in the haystack, but rather to use all the data to characterize the haystack itself. What's its shape? What's its composition? Nutritional value? Things like that. So, for example, with gene expression, as I said, learning about those most important gene networks or pathways that are controlling the dominant sources of variation. If we could do that with our genetics, then I think we would have learned a lot about a system more efficiently and interpretably. So I've been working on a method that tries to address both of these problems, which I'm calling BSFG, or Bayesian Sparse Factor Model for Genetics. And the goal of this is to be a scalable and interpretable extension to the linear mixed model for these big traits. Typical or traditional multi-trait linear mixed model tries to sort of brute force find associations between all genotypes and all traits and trait combinations at once. What BSFG does is it first uses the full data to learn modules or learn groupings of these traits that are sort of the most important in the data, and then does the genetics on each of these ones separately. Other models try to do this, but what I've been trying to do in particular is do this all simultaneously. So we can learn the modules as we're learning the genetics all at once and take advantage of both types of data. So while still maintaining that full power and flexibility of the linear mixed model. The details of how this is done, but it does seem to work, at least as I've tried it. It's capable of doing genetic analyses with 10,000 or more traits in a reasonable amount of time. Now, this doesn't come for free. The way that I'm able to do these analyses is that I'm inputting prior information on what I think these modules should look like. And this may sound kind of scary, like we're making things up, but it's really the same principle that genomic prediction is using today. You use prior information. However, this prior information really can be pretty vague. So in most cases, all I input is the idea or the expectation that each of these modules that it identifies should only involve a smallish number of traits. That's basically what we mean by a module. And then only a small number of these modules are really important. And that's all that it really needs. However, part of the flexibility and power of a Bayesian approach is that if there is more information, say there are known gene networks, you can input those, and the model should become even more powerful. So the output of this model is quantitative genetics in any sense you want on each of the original traits, QTL loci, heritabilities, things like that. But probably more importantly, quantitative genetic parameters for these modules themselves. And so I think that's maybe where the interpretation can come from. To show you this in action, I've been working with Cody Marcos of the Maloof lab to help analyze a data set they generated, using RNA-seq to look at the response of Brassica rapa to growing in competition. So in this experiment, they grew 124 genotypes of Brassica rapa from a mapping population in crowded and uncrowded conditions in the field, and then used RNA-seq did RNA-seq on replicates, on 3 to 4 replicates of each genotype in each of these conditions. And their goals here were to identify genes and pathways that are involved in this response to crowding, and if any of the activities of these pathways was genetically controlled or variable in this population. So it's well known that crowded plants must compete for light, and they do this by a typical shade avoidance response involving elongated petioles and internodes. And then a lot of this is regulated by various light signaling pathways, which are fairly well characterized. And so to test out this approach, we selected, or Cody selected, 170 genes with known roles in the light signaling pathways. And so then we applied this model to this data set with 800 individuals and 170 different traits or characteristics. And these are some very, very preliminary results. But so what I'm showing in this heat map is the estimates from the model of the genetic association or genetic correlation among all 170 traits in pairwise fashion. And so, as we get more and more red, that's a stronger and stronger genetic association among these traits. And, there's a lot to look at here. But I think, by itself, this is a fairly, novel result in that pretty much no other program could try to analyze this much genetic data at once. But I think probably more importantly or interestingly is that we can explain all of these correlations by just 10 important modules or groupings of the genes. And so here I'm showing how many genes are involved in each of these modules. And the first one involves actually most of them, but the others are fairly sparse. And so I think that we can try to start associating those modules with pathways in the network and hopefully get some intuition underlying them. But for now, I want to show that for each of these modules, not only we get its identity, but we get something about, or we can immediately do genetic analyses on them. So here, what I'm showing is for each of these 10 modules, its heritability. How much of the variation in this set of genes can be explained by the genotype it aligns. And mostly they're fairly low, but non-zero for a lot of them. And especially module 4 here is strongly differentiated among the lines. So that's something that we can learn. At the same time, we can ask how environmentally responsive are these modules. And the evidence isn't very strong for a response in any of them, but in particular module 1 and 5 are sort of approaching significance in the sense of a consistent response in response to the shade in these suites of genes. But I think most interestingly, because seed production, or the total seed production was measured on each of these lines in these same conditions, we can also test for genetic associations or genetic correlations between seed production and these suites of genes too. And we find that 4 of them have quite strong genetic correlations between seed production and the suites of expression of these genes. And so I think that trying to follow up on these suites of traits might give us more biological insight into what's controlling overall performance in these lines. So to summarize, what I'm trying to show you is that quantitative genetics, as we understand it, can be applied to these big traits. And doing so might provide important insight into mechanisms and also potentially could be used for breeding. But maybe more importantly, analyses that can be more powerful and more informative if they focus on identifying these underlying patterns in the big data rather than trying to brute force look at every individual trait themselves. And while I've tried to emphasize how this model could be used for very big data sets, I think that it should be fairly effective also for even small data sets with just a few traits. And so if that interests you, I'm hoping to have a pretty usable and accessible R package out pretty soon. It'll be available on GitHub. So with that, I'd like to acknowledge people in my lab, in particular Lexi, a master's student who's been working on developing and implementing this model. Cheyenne Mukherjee at Duke has been a longtime collaborator on this. And then Julien Coley, Steve, and Nan for data, and Steve Knapp and Mitchell for some discussions on this topic. Thank you.
Do you guys have any question?
Could you describe a little bit more how the modules are formed or how you determine the modules?
They are identified based on correlations in the data. We can put prior information on which things we expect to find, but it's really looking for suites of correlations among genes.
So like within that heat map.
It's a correlation. It's a supervised learning method for identifying suites of correlations. But those correlations are the types of correlations that we emphasize are the ones that are associated with the genetics that we're looking for.
Any other question? Thank you.
Yes, Paul?
Okay.
So I think that you rely on RNA-seq to establish these correlations. Is that right?
Well, in the data I showed.
So I'm wondering how much the environment will affect the RNA-seq and how will that affect your eventual result and in the expression of these traits, and especially from a plant breeding standpoint.
So the goal of everything that I'm doing here is that you should be able to analyze any of the data, any of the traits that you're putting in, whether it's RNA or plant height or size, by specifying the environment. So if environment is a treatment, you can measure that. If environment is growing in this location versus the other one, just as you would for yield, you put in field or block or plot in your model, and it accounts for that variation. We can also put in genotype by environment and account for the variation there. But one of the things that models like I'm using are especially good for is using all of the data to identify correlations that can be caused by unknown environmental factors. There's a structure to the data. In RNA-seq data, you always see this batch effects, line effects. These types of things can be very effectively accounted for by this model, and the fact that I'm doing it all at once really makes it so that it can identify those things, account for them, subtract out that variation, and look for genetic variation in the remainder. So I think it should be actually quite effective at accounting for this type of environmental variation, but that's something we have to explore more and test.
Any other question? Thank you, Dr. Daniel. We invite Dr. Alan Ross. He is a faculty member in the Department of Molecular and Cellular Biology. Dr. Alan is investigating the effect of introns on gene expression in plants using the molecular genetics. Please welcome Dr. Alan.
Thank you. So I'm interested in how it is that introns, or at least some introns, are able to have a large positive effect on gene expression. I'd like to start today by going through a lightning review of what we know about the sequences that are important for regulating gene expression. So we know that promoters, of course, are important in regulating expression, and by the broadest definition, a promoter includes any sequences that regulate the expression of a gene. We often think of, we're fairly safe if we start at the start codon of translation and go a kb or two upstream of that, we're pretty confident that we will include all the regulating expression, because this is where the transcription factor binding sites are that determine where and when a gene will be expressed. In addition, of course, we know there are enhancer elements that regulate the expression of some genes, and these can operate in either orientation over very long distances, and so they can be upstream of a gene, downstream of a gene, or in the middle of the gene. And they are thought to help recruit the transcription machinery to promoters to activate expression from them. So work together with promoters. So how do introns fit into this? Well, I'm going to tell you 3 brief stories today. First, I'm going to show you some results that we got asking where within the gene does an intron need to be in order to boost expression. I'm going to show you some information about the relationship between introns and promoters, how they work together. And lastly, sort of returning to the issue of where introns need to be, is the question of whether or not the intron needs to be in the DNA that's being transcribed in order to boost expression, or whether it operates from the RNA that's made. And likewise, whether or not the sequences even actually need to be within an intron at all, whether intron stimulating sequences can also work outside of the context of an intron. Most of the results I'll tell you about today were with a strongly stimulating intron from a ubiquitin gene, which I've put into a reporter gene, which is a fusion between the Gus reporter and a biosynthetic gene from the tryptophan pathway. I'm measuring expression as mRNA accumulation, and the numbers that I'll show you reflect the degree to which steady-state RNA levels are increased by the presence of an intron relative to an otherwise identical intronless control in single-copy transgenic Arabidopsis. I'll also show you some pictures where enzyme activity is visualized by histochemical staining for Gus enzyme activity. Okay, so where does an intron need to be in order to boost expression? We've tested this intron in a total of 14 different locations scattered throughout our favorite reporter gene. And I'm just going to show you the most important results. And that is, of those 14 locations, the intron only boosts expression from 6. So this just shows, again, a diagram of the reporter gene. This rightmost arrow represents the major transcription start site for this gene. This gene has several transcription start sites, but a variety of techniques indicate that this is the most commonly used one, and this is the first known transcription start site, this arrow on the left. What we can see from this Norman blot is that this intron only stimulates expression when it's in one of these 6 locations here. If it's in position 5, like upstream of the transcription start site or further upstream, it has no effect on expression. Likewise, if it's at this position or further downstream, it also has no effect. And by the way, in this Norman blot and the other plots that I'll show you, if you see more than one lane under the same heading, that just represents the results from an independent transgenic line containing the same construct. So these introns are unusual in that respect. They're unlike enhancer elements in that they only work over relatively short distances. So in other words, this intron can boost expression from more than 500 base pairs downstream of the start site, but not twice that far. So that's the first story. The second question is, what's the relationship between introns and promoters? And I'll show you 2 lines of evidence that the introns don't need prior promoter activity in order to boost expression. So one line of evidence is that the introns are able to override the tissue specificity of promoters. And so this just shows results from 2 different genes. So again, these are different Gus fusions here, fusions to different genes. And for each of these, I'm showing you the results from 3 different transgenic constructs. One which contains the gene's endogenous first intron, one contains no intron, said that intron removed, and in this case, the intron has been replaced with that stimulating ubiquitin intron. And the point is that this intron can strongly activate expression in tissues in which those promoters are normally inactive. This is just a northern blot from the publication first describing this gene to show that these transgenic constructs really do reflect the fact that this gene is normally not on at all in roots. So introns can override that normal tissue specificity. Perhaps an even more striking example of the independence of introns from promoter activity came from experiments we did in which I was trying to construct a negative control from experiments I had planned. Again, this is back to the TRP1 Gus fusion here. So to cripple the expression of this gene, I removed all the sequences between positions 5 and 3 here. So there's 300 nucleotides, contains all the known transcription start sites, all but 18 nucleotides of the 5' UTR. And I was startled to find that that made no difference to the expression of this gene whatsoever. So this again is a northern blot containing the fusion with the promoter intact or the promoter deleted. And if you pay attention to this lane here, so this is the construct containing the ubiquitin intron to show some degree of how well the intron is expressed. Deleting the promoter basically caused no decline in the expression of this gene. So if you ask yourself, which is more important for regulating expression, introns or promoters? Well, this construct contains both. If you delete the intron, expression drops more than 90%. And if you delete the promoter, nothing happens, or maybe expression even goes up slightly. So if you delete all the transcription start sites, where is transcription starting? And this is a little bit more detailed picture of this gene, of the endogenous TRP1 gene, with all the transcription start sites as determined by a variety of different methods mapped. And again, this is what I was considering the major transcription start site and the first known transcription start site. In the constructs, our transgenic constructs containing the ubiquitin intron with the promoter deleted, we used 5' race to map the transcription start site and found the transcription was starting 300 nucleotides upstream of the normal transcription start site. These sequences are aligned to the endogenous TRP1 gene, so the deletion shows up as a gap here. But of course, there's not a hole in the DNA. If you close that gap, you can see the transcription is starting basically the same distance upstream of the intron as it normally does. So it seems as though the intron does not require an active promoter to boost expression. It's somehow causing transcription to start upstream of itself. Okay, the question is whether or not the intron boosts expression when it's in the DNA that's being transcribed or from the RNA that's produced. I have some evidence that it's acting at the DNA level. So the idea here is that I made a series of hybrid introns between this ubiquitin intron that strongly stimulates mRNA accumulation and this very similarly sized intron from a cold regulated gene that has very little effect on expression. So in these hybrids, I kept the ends of this of the introns from this cold regulated gene in their normal orientation to allow for intron splicing, and cloned in between those ends the middle portion of the ubiquitin intron in either orientation, and found that both of these introns stimulated expression equally well, despite the fact that the RNA produced by these two is very different. It's the reverse complement of each other. So this is much easier to explain with a DNA-based model of intron action than an RNA-based one. Second question is, so we've been able to identify intron sequences that stimulate expression and can use those to turn an intron that normally doesn't increase expression into one that does. And the question is whether or not those sequences need to be within an intron to affect expression. So we're able to identify, again, in our TRP1 Gus fusion, we identified 6 locations that had the proper nucleotide composition that we could rearrange these nucleotides just by shuffling them around to create a match to our motif and ask whether or not that it was able to stimulate expression. So again, this is northern blot. So this is the motif that we were able to create those locations, again, without creating too much of a change to the encoded amino acids. So this is the motif that, again, this is identified in collaboration with Ian Korf and his group. So we introduced either that motif or a mutant version of that motif in which the central 2 nucleotides are reversed, which we'd shown previously virtually eliminates the ability of this motif to turn an intron into a stimulating one. And you can see that intronless constructs containing 6 copies of this motif encoding sequences stimulate mRNA accumulation almost as much as do the 6 copies of that motif in the presence, in the context of an intron. So from this we conclude that these sequences do not need to be in an intron in order to boost expression. They seem to be able to stimulate expression from the DNA context as well. So just to summarize what I've shown you then, these introns have fairly strict location requirements that need to be transcribed within the first kb or so. They don't seem to require the activity of a promoter. In fact, they don't seem to need a promoter at all. And it looks as though they're operating at the DNA level and don't need to be in an intron to do so. So just to tie this back into what I went over at the beginning, let's see how this fits into this scheme. You know, how do introns compare to these? Well, I've shown that introns don't need to be upstream of the ATG. They can operate from several hundred nucleotides downstream of the start codon. We have no evidence that transcription is starting in the intron. At least we've never seen examples of transcripts that map to within the intron. And so we think it's therefore unlikely that it's binding general transcription factors. Whether or not they bind regulatory transcription factors is an open question. But if they do, these must be unusual transcription factors in the fact that they can activate expression almost at more than 500 nucleotides upstream of themselves. I've shown you that this intron does not stimulate expression from upstream of the gene or downstream of the gene. It is intron independent, orientation independent, but it doesn't function over long distances. Again, I would call it mid-range, more than 500 nucleotides, but not more than 1,000. And last of all, they don't seem to need a promoter in order to do that. So my conclusion then is that these are the almost total lack of correlation between an intron and these known regulatory sequences suggests that introns are doing something different. They're a kind of regulatory element that we aren't used to thinking about. All the work I told you about today was done by myself or my graduate student, Jenna Gallegos, with some help from some undergraduates and technicians. And we're grateful to Ian Korf and his lab for doing the computational work, and especially Shram Amami, who did some work for the tissue-specific expression. I've had funding from the USDA and NSF over the years, and Jenna has had various scholarships and fellowships from these three funding agencies. And I think I'll stop there, and I'll be happy to answer any questions.
So you show that if you delete the promoter, right, you still get activation at sort of the equivalent transcriptional start site.
Yes.
And if you have a promoter and you reverse the orientation of the intron sequence, then it still works. So if you delete the promoter and reverse the orientation of the intron, where does the transcript start?
We haven't done that experiment. We have not deleted the promoter and flipped the intron around backwards. We have shown that there's a limit to what sequences you can delete and still have expression. If you delete all the way up into the tail end of the upstream gene, those constructs are as dead as we thought the first ones would be. Yep.
I was just wondering if you knew if those motifs are associated with any, I mean, you said that there was no transcription factor, but if it maybe is associated with things that might be opening up chromatin or regulating the transcription, yeah, as you said, more generally.
Yeah, so let's say they don't match anything, they don't perfectly match anything in the known databases of known plant cis-acting regulatory sequences. I like the idea that they open chromatin structure. I think that's plausible. To me, it's almost easier to think of something acting upstream by opening chromatin, like, you know, full of nucleosomes. It's somehow easier for me to imagine than distances of 500 base pairs in the linear DNA. But so yes, I think it's a viable possibility. Yes?
Do you think your results are applicable to most ubiquitin introns, or do you think this is very specific to the ubiquitin 10?
So yes, I didn't mention that. So all the results I showed you were with the ubiquitin intron. For virtually all of the results, we've found those also with other introns. Yeah, other than the ubiquitin introns. Ian Korf generated an algorithm that can predict on the basis of sequence whether or not an intron is likely to stimulate expression, and ubiquitin introns very often score high in that algorithm. Yeah, quite often. So we think of introns as being, I think of introns as being the brick on the gas pedal. They are good for driving genes for which it's important to have a high level of expression throughout the plant all the time. Unlikely to be found in genes that are regulated in some way. They only expressed in some tissues at certain times.
Yes. Have you looked at testing multiple introns in different locations rather than one intron?
Could you repeat the question, please?
Have you tested multiple introns, one like within the promoter just before the ATG and one in the to see what is the effect on multiple introns?
Yes, so have we looked at multiple introns at the same time in the same gene? We've done a little bit of that, but not very much. I think there's a lot that could still be learned about that. One complication is the fact that the distance dependence seems to be, then you near the beginning of the gene in order to affect expression. If you start putting in a second intron before that, then you've also moved it further away, and it might not work as well anymore.
Any other question? Thank you, Dr. Ross.
Okay.
Now, I would like to invite our last speaker from UC Davis faculty, Dr. Siobhan Brady. Dr. Brady received her PhD at the University of Toronto in 2005 and was a Natural Sciences and Engineering Research Council of Canada postdoctoral fellow at Duke University from 2005 to 2008. In 2009, she began an assistant professor position and became an associate professor in 2015 at the University of California, Davis in the Department of Plant Biology and the Genome Center. Her lab focuses on the global regulation of gene expression and its contribution to root morphology and development in Arabidopsis thaliana, Solomon species, and maize. In 2016, Dr. Brady was named as a Howard Hughes Medical Institute faculty research scholar. Please welcome Dr. Siobhan Brady.
So can everybody hear me? Yep. Okay, so thank you. I'm really happy to be here. And this has been also an exceptionally organized conference on behalf of the students. I've never really seen something so well organized in my life. So kudos to you. Today I'm going to speak to you very briefly about one particular research project in my lab, which is titled Regulation of Plant Development in Response to a Changing Environment. So this infographic was published in the New York Times last year. And it shows the number of days that are predicted to be above 100 degrees Fahrenheit across the United States. And so for those of us in California who are in the Central Valley, we're very familiar with the extensive amount of heat that we've experienced, the 7-year drought. As an example of what's associated with this 7-year drought is, of course, the complete lack of rain, right? As an example of how profound that can be, my 4-year-old, who just spent 3 months in the Netherlands, was terrified when he saw rain regularly and did not understand why you would ever need a winter coat or mittens. So there's that. Also, so there's a predicted huge increase in the number of days above 100 degrees Fahrenheit in the southeastern US, which is also associated with an extreme in availability of water on the other end, which is a huge amount of water due to flooding. Oh my goodness, all my backgrounds are off here. In my lab, we're interested in phenotyping across scales. So geographic scales, which are represented in terms of extremes in water availability. And so we're interested in looking at root system architecture, which was beautifully introduced by Chris Topp, and how root system architecture changes in response to a lack of water in terms of drought or an increase in the amount of water in terms of flooding. We're also interested in exploring both within and between species, and this is part of a really large 4 research group project that's being funded by the NSF Plant Genome Research Program. And so collectively, we're looking at 4 different plant species. So Medicago truncatula, which is looked at by the group of Roger Deal at Emory University. Rice, which is looked at by the group of Julia Bailey Serres. And Solanum lycopersicum, a domesticated species of tomato. And Solanum pinellii, which is a wild species of tomato, which is drought adapted and drought tolerant. And this is looked at, my group looks at the root system architecture, and the group of Neelima Sinha looks at the contribution of shoot meristems. So we're also interested in exploring the response of the root system at the level of individual cell types, because it's been shown that signaling and development within individual cell types can control growth and adaptability in response to these extremes in stress. So my group has, and this is primarily done in work established by Kaisa Kayala, a postdoc in my lab. She has generated a cell type atlas in tomato as just an example, and she has generated 11 different cell type or tissue specific promoters which drive expression in individual cell types or tissues, in this case within the cultivated tomato cultivar M82. Now collectively as a group, all 4 of our research groups have worked together to identify and establish promoters which mark cell types in a similar manner across these different species. These include constitutive promoters, promoters that drive expression in growing tissues or in meristem tissues, promoters that drive expression in 2 different regions of cortex cells within the root, in the stem cell niche, as well as in the endodermis and in the vasculature. So we are further interested in profiling different suites of molecular signatures at cell type resolution. And so we use 2 different technologies which had previously been established in Arabidopsis by 2 different members of our group. And we have now translated these to each of these 4 different species. The first technology is called intact or isolation of nuclei tagged in individual cell types. In this particular case, for instance, with one of the 11 cell type promoters that we have found to drive expression in tomato, we take the cell type specific promoter and drive the expression of a biotin ligase. We're then able to immunopurify those those nuclei using streptavidin magnetic beads. The second technology is called TRAP, or translating ribosome affinity purification. And in this particular method, we take, again, the same cell type specific promoters, and we fuse them to a flag tagged ribosome. We're then able to immunopurify those ribosomes in a cell type specific manner. So with intact, we're able to further take these purified nuclei and profile 2 types of molecular signatures. The first is using a method called ATAC-seq, which stands for Assays for Transposase Accessible Chromatin. In this particular method, we expose the chromatin within these nuclei to a transposase, which is able to then go and cleave regions of open chromatin. So in this way, we can get very specific regions of chromatin availability that are likely the important regulatory elements. Alternatively, we can take these nuclei and we can purify RNA. Also, we can specifically purify RNA that has undergone a variety of forms of RNA processing. With the TRAP methodology, we're again able to look at 2 different molecular signatures. In the first, we can look at mRNA that has been associated specifically with the ribosomes as a proxy for transcripts that are undergoing translation, or as a proxy for translation. The second method we can use is called RiboSeq. With RiboSeq, we're able to look at very specific regions of RNA that are associated with ribosome that are essentially footprinted by the ribosomes. And this can provide evidence of an additional form of potentially translational control of those transcripts. So again, work done by Kaisa in my lab. She has now established an atlas of 11 cell and tissue types for these different intact lines and for these different trap lines in Solanum lycopersicum, this cultivated variety of tomato. And then she's also established a similar atlas of 6 different cell and tissue types of this drought-adapted or drought-tolerant species of wild species of tomato, Solanum pinellii. Okay, so what type of information can we obtain using these particular lines in response to these changes in water extremes? And so collectively, our 4 groups have established conditions in these 4 different species that allow us to look at submergence stress from the same physiological perspective. And so we determined the right time point to profile roots in response to submergence stress by looking at expression of the alcohol dehydrogenase gene, which is shown here in blue relative to the controls in green. So I'm going to show you an example of how we're able to look at this regulation using several different tracks. So in this first track, so again, this is all data from rice taken from Julia Bailey-Cereza's lab. Here we're looking at the gene model of this alcohol dehydrogenase on the bottom of this plot. And then in this first two rows, so it's always arranged in this way, the first row are roots that are grown under standard conditions with sufficient oxygen. Whereas in the second row, you can see roots that are exposed to oxygen stress or submergence. They're grown under anoxic conditions. So we're looking here at chromatin availability using the intact profile, using the intact ATAC-seq profiling. You can see an increase in the number of peaks here corresponding with the promoter and the 5' UTR regulatory region of the alcohol dehydrogenase. Next, we can look at mRNA. And you can see that the mRNA from intact the mRNA with intact is showing increased expression only in the anoxic or submergence conditions. This is looking at total RNA, which somewhat mirrors the nuclear RNA. And then here you're looking at trap RNA. So this is RNA that's associated with the ribosomes. You can see that this very closely mirrors total RNA, which likely reflects the fact that most RNA isolated from total RNA is largely cytosolic RNA. And then at the very bottom here, you can look at these ribosome-protected fragments. And under anoxic conditions, you can see that there are fewer ribosome-footprinted fragments at the 5' UTR, which is what you would expect, as this particular region will not be undergoing translation. This is an example of a type of snapshot that we can obtain just for a single gene in a single species. Another type of data that we're able to obtain is a transcription factor footprint. So with these ATAC-seq reads, or these regions of chromatin availability, what you can do is you can look at the peak that results from this ATAC-seq read, and right in the middle of these reads, you can often identify a shadow in these reads. This shadow represents a region where a transcription factor has bound and is protecting that particular region within the open chromatin. Here I'm showing you a set of footprints which Roger and his group has identified from Medicago from about 580 open chromatin regions that are open specifically in response to submergence. They were then able to identify over 2,000 different transcription factor footprints, which actually represent the output of transcriptional regulation in response to submergence. So what type of novel biological information can we obtain using these approaches? So the tomato submergence response has never been characterized before, and Kaisa went and further looked at a suite of genes in both the intact and trap RNA-seq data sets that are enriched in response to submergence in both the domesticated tomato species, M82, and also in Solanum pinelii. Here she found a group of genes enriched that are associated with the core hypoxia response as determined by looking at Arabidopsis data, which shows a nice conservation of this process between Arabidopsis and tomato. Furthermore, when we look at the transcription factor footprints, we see an enrichment of the known hypoxia response element, again indicating that there's likely conservation at the level of cis regulation for this particular response. We also identified suites of genes that are associated with the response to a variety of different stresses, as well as genes that are associated with developing xylem development, which we're still not certain what the biological significance is of that. Gene categories that are underrepresented in response to submergence include a variety of categories associated with dampening of both translation as well as energy, which has also been well characterized for the submergence response. So now I'm going to switch very quickly to speak to you about work that we're able to do to look at these responses to changes in water extremes at the level of individual cell types. So 90% of flowering plants, it turns out, have an exodermis, but Arabidopsis thaliana does not, which means that we know very, very little about exodermis development. And so the exodermis is this outer layer of cortex cells. And again, most plant species actually have multiple layers of cortex cells. So this outer layer of cortex cells is known to undergo lignification and suberization, although the developmental mechanisms and molecular mechanisms underlying this are unknown. So we have been able to look at tomato root system architecture and exodermis differentiation in response to these extremes in water stress in a domesticated species. And you can see in response to waterlogging, there's a huge increase in the number of adventitious or shoot-borne roots compared to the other conditions. We're also able to see a very specific change in the levels of suberin within these exodermal cells, shown here only in response to drought. And we also see a change in suberin deposition in the pericycle cells, or the cells that are flanking the vascular cylinder. We're further able to compare these changes in between different species. And here you see suberin deposition in tomato and rice. You can see, again, the huge accumulation in the exodermis in response to drought, but a very small amount of deposition in rice. And then if you then compare this again to waterlogging, we see deposition in the exodermis layer as well as the underlying cortical layer. So we see changes in response to different extremes of water as well as changes in cell type specificity depending on which species. And so we're further able to now go and use our cell type atlas data and actually further characterize the developmental mechanisms that are responsible for exodermis development and lignification and suberization within this particular species. So very quickly, I'm going to just highlight the fact that we're also able to see other changes in cortex layer development that have never been reported. So for instance, in response to waterlogging, we see a huge increase in the number of cortex layers within the root, as well as an increase in the porosity between these cortex layers in response to flooding, which are not actually a parenchyma, but we predict may function as a parenchyma. Okay, so finally, I'm finished. I'd like to thank all of our collaborators, including the groups of Julia Bailey-Serez at UC Riverside, Nilima Sinha at UC Davis, and Roger Deal's group at Emory University, and Kaisa Kayala did most of the work in the lab, including Niba Nirmal, who's here in the audience, and Reina Sands. Funding from NSF, Plant Genome Research Project, and my HHMI funding is being used to complete these cell type atlases. And so thank you, and I'm happy to take any questions.